pleasure to be here, and thank you to the organizers for such a wonderful meeting. It's been great. And like Peter said, I'll be presenting a tale as old as time, Beauty and the Beast of the Ocular Surface. I have no financial disclosures. And before I begin, I really want to acknowledge the authors of the story that I'm essentially just really narrating. And that's members of the UL1 Penn Group, specifically Tariq, who is a wonderful graduate student, Huasheng, who's a great postdoc, and Noor, who's a great postbac. I'm going to stop saying great because they all are great. So instead of being repetitive, and they're from the Low Lab, from the Rompalis Lab of Hisuk, who is a postdoc, and Chi, who is a postdoc in the Wu Lab, as well as members of my lab, Yoka, who's a senior research investigator, Kate, who is our postdoc, and Chenyan, who's our senior research technician, as well as our really talented undergraduates, Chris and Kim, as well as our financial sources, because without their support, there's not even a story to begin with. So for those of you guys who are familiar with the story, The Beauty and the Beast, it takes place in a small provincial town in France. But this time, in this story, this town is called the Cornea. There's 5 territories here in the Cornea, and we're really going to specifically stay up in this northern area. Here's the laser pointer. So we're going to really stay in the northern territories, while my friend and colleague, Dr. Rompalis, will be staying here in the southern territories. And the function of this town is really 2 major functions. It's to protect the inhabitants of the town, as well as to refract the light and bend the light so it reveals the truth. Some of these inhabitants in this northern territories are the corneal nerves. They play a really essential role in homeostasis, but also in disease processes. And if we remember back to how the Beauty and the Beast story starts, it starts with this sorceress who comes and visits a very cruel-hearted prince. And because she was disguised as an old lady, he sort of treated her really badly. And so she curses, right, his entire kingdom and turns him into a beast. So in this town of the Cornea, it's also cursed with many corneal diseases, and unfortunately that affects the corneal nerves. And one of the disease processes that we decided to focus on for the UL1 group is dry eye disease because of its prevalence. It affects about 16 million Americans and about 5 to 35% of the global population. There's really 3 general causes, very sort of broad, that cause dry eye disease. Evaporative, where it's really the lipid layer that's deficient. Aqueous, where the watery layer is deficient, and where most of the patients lie, which is the sort of a mixture of both of these causes. Symptoms include redness, hypersensitivity, itchiness, and pain. And ocular comorbidities include decreased quality of life, loss of vision, degeneration of sensory afferents. But what was really particularly interesting for us is that there's a cohort of patients who actually report both insensitivity and hypersensitivity in DED, suggesting different responses by neuron subtypes. So in the story, it's the prince that gets sort of cursed and turns into a beast. But as the story goes along, we realize that there's more than just one beast in this story. Just like there's multiple subtypes of corneal nerves in the town. And so most of them are polymodal nociceptors, which is imaged here by the Rompalis Lab. This beautiful image of a TRPV1 Cree confetti mouse, which highlights individual afferents with different colors depending on the recombination of the reporter genes. Most of these are TRPV1, so they're mostly the nociceptors, and they build up this like beautiful concentric circle that we appreciate clinically on confocal microscopy. Then there's also the mechanoreceptors, which is about 20%, and then the cold sensing thermoreceptors, which is about 10%. So the question that really you want to ask in the Beauty and the Beast, right, is like, are all of these beasts alike, right? Do all of these nerve subtypes contribute equally to epithelial homeostasis and disease? So that's the real question. There's multiple approaches, aka point of views, if we're keeping up with a literary analogy, to address this first question. So the fundamental sort of question that we had for our group are the sensory neurons fundamentally equivalent, or do they acquire distinct tissue-specific identities? And so I'll present like sort of 3 point of views here. And so with this first point of view, we decided, can we distinguish these corneal sensory afferents morphologically? And so we employed a combination of mouse lines with immunohistochemical staining to either genetically or immunohistochemically label the different subtypes of the corneal sensory afferents to really assess their distribution and morphology. So here you can see that we sparsely labeled the avalon-Cree ERT2 inducible line. For the dermal receptors, we did TRPM8. For the low threshold mechanoreceptors, we did neurofilament heavy immunohistochemical staining. For our nociceptors, TRPV1, Cree, AI9, as well as CGRP stain. And then this last group, which is really interesting to us because it's sort of a group that wasn't really established in the cornea, which are the somatostatin expressing peripheral sensory neurons, which we use the SST Advilin-Flippo AI65 mice. And then obviously beta tubulin as our sort of broad denominator that stains all the corneal nerves. And so this is work that was performed by our wonderful graduate student, Tariq, where he looked at sort of the distribution and the morphologies of these different subtypes. And we could see for the thermoreceptors, the TRPM8 positive cells, that they sort of start out in the periphery and then kind of as like these medium-sized branches, and then immediately start to branch into these finer branches in this sort of I keep saying sort of, in this alternating pattern of, sensory afferent, space sensory afferent, which we like to refer to as the peppermint candy sort of pattern. And this is different from our neurofilament heavy positive corneal sensory afferents, say that 5 times, which are usually more limited to the peripheral area, and there's sort of bigger branches, doesn't have that fine branching that you see with the TRPM8, and they stay sort of more in the periphery, not so much projecting towards the center. And then we have our CGRP positive corneal sensory afferents, where you have those medium branches, but they make sort of more of like a crown around the periphery, then finally branch into the center. And unlike the TRPM8, where you saw that alternating pattern, here it really covers almost all of the cornea and really forms much of that sort of swirl. And then last but not least are our somatosensory positive. Somatostatin. I was like, there's something wrong with that. Somatostatin positive corneal sensory afferents, which morphologically looks a lot like the TRPM8 positive as well as the CGRP neurons. However, they really only occupy at most a hemisphere of the cornea, and most commonly they at least hit the quadrant of the inferior nasal area, which kind of makes sense with the hypothesis of what it functions in the cornea. It's immunomodulatory, and so it receives in that location probably a lot of signals from the tear film because it drains through the puncta right there. So at least we could see on a population level this sort of separation of these distinct subtypes, not only where it sort of is distributed in the cornea, but also morphologically how they look. So then the second point of view that we sort of asked ourselves is, can we distinguish these subtypes transcriptionally? So we all know that the soma bodies for the corneal afferents exist in the trigeminal ganglia. They send their afferents to the cornea, but then they also project to the brainstem. But the characteristic of the soma bodies in the trigeminal ganglia are really unknown. It's unknown what their identity is. It's unknown how heterogeneous the population is. And mostly, most of the challenges is because there's not a really great way to isolate these corneal afferents and retrograde label them back to the trigeminal ganglia. And so this was a big project of the UL1 group, and our very talented postdoc Hisuk developed this intrastromal delivery of AAV retro for targeting the corneal afferents. So he would literally put or pierce through right at the center of the corneal stroma. This is like extremely thin, and then just deliver about like 1 microns of volume in the stroma. And then you can see retrograde labeling of these cornea-specific neurons in the trigeminal ganglia. He harvests the trigeminal ganglia in 28 days after the injection. And oddly enough, it really is limited to this very specific geographical area in V1. We weren't even sure if there could be even some labeled outside of V1, but it seems pretty specific for this area. And depending on, and if I could get this video going, because it's a beautiful video that he's recorded, you can see all of these labeled neurons. And surprisingly, if you increase the volume in the intrastromal injection, you can actually highlight even more labeled neurons. So we estimate there's about like 300 of sort of corneal sensory afferent neurons, or corneal sensory neurons in the trigeminal ganglia. And so in order to do sequencing, he did LCM, or laser capture microdissection, which you can see here in the cartoon schematic. Here's the soma that is prior to laser microdissection, and then after laser microdissection. And then we physically isolate these individual fluorescently labeled neurons. And what we see is that they sort of resolve into distinct transcriptional clusters. You can see here on the left-hand side, the non-labeled, or our control, as well as the retrograde labeled, have similar characteristics, similar median number of genes per cell, similar median transcripts per cell, but they cluster into these 9 different categories. And this is when you start to see a distinct sort of difference between the labeled and the non-labeled. And this was in collaboration with Huasheng and Qi from the Wu Lab and the Lo Lab. And you can see that probably the most visually apparent one is cluster 0, way more presented in the labeled neurons versus the non-labeled. And then when we look at the differentially expressed genes, we can see that there's a unique transcriptional programs for at least the labeled, the corneal sensory afferents, versus the non-labeled. And we can see here in the green, I know the type is really small, but here in the green, we can see the genes that are upregulated in the labeled, and then also in the non-labeled. And in these top genes that are upregulated, we can see that there might be mechanistic reasons for them in the corneal sensory afferents. And we have a nice positive control where we have the ROSA 26 locus here only in the labeled area. So in the third point of view, we wanted to know, how do these corneal sensory afferents respond to pathological conditions? And so with our help of our very talented postdoc, Kate, we used an aqueous deficient dry eye model. There's numerous reasons as to why we chose this model. There's always pros and cons to all models, but this is sort of a clean model that we decided to use. And it requires excision of the extraorbital and the intraorbital lacrimal gland. And then you develop signs of dry eye disease that lasts actually months after the procedure. And you can see here some neovascularization and some haziness to the corneal surface versus the contralateral eye, which was not our control, but we used a sham in separate mice in the right eye for the control. And when we look at these subtypes, we can see that there's a remarkable difference in the response by subtypes. So here at 4 weeks, I have an example of the TRPM8 positive cells. And we can see at 4 weeks after dry eye or lacrimal gland excision, you can see that the peripheral sort of branches get bigger, and then there's dropout in the central area versus the CGRP positive cells. We can see 8 weeks after excision, much more bigger branches as it goes towards the center, and a really sort of messy network of these nerves centrally. And then 8 weeks for the nerve, for the NFH positive cells, we can see a little bit more projection into the center, a little bit bigger caliber of the nerves. But what was most astounding is that in the somatostatin positive cells, we can see absolute dropout of these nerves after dry eye disease, where if you think about if it's a immunomodulatory function, it kind of makes sense because dry eye disease is essentially an immune process. And when we look at the it's miswritten, it's sequencing, but it's actually qPCR. We can see these maladaptive reprogramming in these nerves where you have loss of normal functions, such as sensory impairment, loss of protection, regeneration failure, and reduced inhibition regulatory mechanisms. an abnormal gain where you have mechanical hypersensitivity, aberrant signaling, and disorganized structural expansion, which goes really nicely with what we found morphologically with genetically labeled subtypes and the immunohistochemical staining. So it shows that dry eye disease disrupts the sensory and the protective regulatory balance. And so it gives you a clue into sort of the characteristics of these different beasts. So where are we now? We know that there's multiple corneal nerve subtypes. We also know that there's a new category that's really interesting for us with the SST positive nerves. Our goal is to further define their identity and then their heterogeneity, link them to their disease-associated reprogramming, where we have some data and we're mining that even more. And then our next big step is to really correlate these to function as well as behavioral responses. So really taking from identity to mechanisms of pain to therapeutics. And so hopefully, I'm sorry I'm leaving you guys on a cliffhanger, but hopefully the tale is to be continued. And I'd like to thank, again, all of the members of the UON group and members of my lab, as well as our funding resources. So thank you.